Previous lecture. 1. Emphasis on evolution of behavior, adaptation, inferring a genetic basis. Here's a story and until you make up a better one, I win. 2. Emphasis on gradualism, that slow evolutionary change, the molecular mechanisms for gradualist evolutionary change. Microevolution, point mutation, deletion, insertion. Mutations, potentially knocking a protein completely out of business, and you get a different gender than you actually are chromosomally. Micromutations, affecting how readily a protein does its job, how strongly, potently, long. Then. Attacks to gradualism. Punctuated equilibrium. Most of the time, nothing was happening, and then sudden, dramatic change, by Gould. If most of the time nothing happens in terms of evolutionary shift, there goes gradualism. There goes the emphasis of every bit of adaptation is going to make a difference long term, to compete. All that goes down the drain if 99% of the time there's equilibrium stasis. So status and rapid change for paleontologists bears no resemblance to what's like for an evolutionary biologist. They can just see evolution of morphology. Miss the world inside that morphology. And show me some molecular mechanisms for these macroevolutionary changes. Then. The structure of genes and DNA is wrong. There are the intervening sequences. The intron/exon organization of genes. Mix and match combinatorial abilities for one gene, or a stretch of DNA. To specified proteins. Depending on the function of the splicing enzymes. And those splicing enzymes differing in different parts of the body under different circumstances. Then. 95% of DNA. Not coding for proteins. Not being genes. But instead, the instruction manuals, the promoters, the switching on and off. Leading to transcription factors, transducing events going on outside the cell/organism, into changes in DNA. The role of transcription factors, promoters. Their lock and key interactions. And, if microevolution is changing the function of a protein. Changes occurring at the level of transcription factors or promoters. Those are changing the context for which proteins are functioning when genes are expressing. If something happens outside the nucleus, cell. Then, if that results in a gene being activated, you'll see the response. Second thing we trashed. DNA as the dogma of life. DNA, RNA, and protein commands in life. But DNA, genes, are just a readout, and most of what it's about is environmental regulation when genes activate. Finally, genes have nothing to do with the sequences of DNA. But instead, has to do with access of transcription factors to the DNA. Changing accessibility permanently. Epistatic, epigenetic. And fertilization is about genetics. Development is about epigenetics. Where, genes don't really know what's up. In addition to mechanisms know for microevolutionary change. And how these things can set you up for big macroevolutionary changes. E.g. -1. A gene coded for 3 exons. Produce a messenger RNA that encompasses the 3 genes plus introns. Along comes a splicing enzyme. Knocks out the genes. And wind up with a mature protein. So, what if you have a mutation in your splicing enzyme? You get 2 novel proteins. It's not a little microevolutionary change. Not a protein working better or more sluggish. It's the invention of entirely new proteins. The splicing factors working differently, it's a major change. Like, an outside world event happens, to activate the gene. You produce 2 novel proteins. So, if a splicing enzyme, if enzymes are genes. There's a gene for the splicing enzyme. That gene has its promoters, in a number of exons, and this it needs a splicing factor, equals big problems. -2. A mutation in a promoter. Mutation in a non-coding part of your DNA. And run immediately with that. Have a different promoter that will interact with a different transcription factor. The same version of the promoter, multiple copies of that promoter, appear upstream of different genes. Promoters mediating expression of entire network of proteins. Change the promoter equals change the network. Change it in only some of the places where it occurs equals novel network, of half of the proteins. Or the whole network equals different transcription that interacts with the promoter, equals a network of proteins that have never existed before. Novel genes, novel network, novel if/then clauses. E.g., 1. A promoter upstream of a gene having to do with the hormone vasopressin. Vasopressin, hormone associated with social affiliated behavior in males, is a vasopressin receptor, thus there's a vasopressin receptor gene and a promoter upstream of that gene. Male vole. Rodents. Monogamous or polygamous. In monogamous vole species, there's a different promoter upstream of the vasopressin receptor gene than you find in the polygamous ones. Use gene therapy techniques to change the promoter to modify it. And you could convert a polygamous into a monogamous. So, change your promoter dash different pattern on expression dash different parts of the brain wind up dash major shift in behavior dash, no causing a change in a gene, dash changing the promoter dash you've changed a major if slash then clause. Study. Human male. Which version of that promoter you have gives you a predictive power over how stable your social relationships are going to be. So, have a different type of promoter and you're more likely to get divorced. Again, that's not a difference in a gene, that's a difference in the promoter. E.g., 2. A gene that codes for hormone neurotransmitter of pain perception called dynorphin. Related to morphine. A dynorphin gene. There's a promoter upstream. The number of copies of that promoter in different rats predicts how readily they become addicted to drugs. We've now seen changes, subtle or dramatic changes in splicing enzymes. The genes underlying them. Changes in promoters. -3. A mutation in a gene for a transcription factor. Do that, and you'll get a different network. You'll have changed if slash then contingencies. E.g. Human genome vs chimp genome. Sharing 98% of our gene/DNA in common. What are the differences? A disproportionate share of the genetic differences are genes that code for transcription factors. You get a change in a gene coding for a structural protein, and maybe your muscle will bend in a different way. You get a change in the gene coding for transcription factor and you'll have invented novel networks. King and Wilson came up with the 98%. Using primitive molecular techniques. And also came up with a prediction, a theoretical one. The most interesting changes that will occur in evolution are in regulatory parts of DNA rather than in the coding parts for proteins. They are right. E.g., 1. You want to turn an ancestor of human and a chimp into something completely different from a chimp, turn into us. Change stuff with regulation, transcription factors, promoters. This is networks amplifying effects for macro changes. Hunter Fraser. How much evolutionary change is being driven by change in regulatory parts of the DNA world, rather than genes itself? Changing how protein works, that's okay. Changing if slash then clauses for entire network, that's hugely important. E.g., 2. Genomes. Of about 100 different species. Sequenced and ranging from short to large genomes, when you pile them up. And look as a function of how long their genomes are. The more genes you find in a species, greater the percentage of those genes that are transcription factors. You're having an exponential increase in the number of transcription factors you need to take advantage of all the possible combinations of network of gene expression. The larger the number of genes, the greater the percentage it is of transcription factors. Get tiny micro changes in DNA coding for transcription factor, splicing enzymes, promoters. And you're going to have big major consequences. Microevolution, about the function of proteins. Macroevolution, about which proteins, when network if slash then clauses are more consequential. Another domain of. We're not talking about a micro change in one base pair. Barbara McClintock. Plant geneticist. Wildly successful. Member of National Academy of Sciences. At that time. The only way to make sense of what is being inherited. Patterns of inheritance. It was just looking at the phenotype, appearance. She's working on it, and sees a result. A pattern of inheritance which appears in certain circumstances, and came up with the conclusion. The only way to explain how this change was occurring was if genes were picking up and moving around in the DNA. If genes were mobile, that there are such things as transposable genes, transposable genetic elements, transposons. But the general consensus in the field was that Barbara M. had gone out of her mind. She disappeared from the field, working by herself for decades. She wrote papers about it that were incomprehensible. Finally, in 1980s, molecular techniques caught up enough to show she was absolutely right. Now called transposons. Transposable genetic elements. Genes actually move. They gave her Nobel Prize, in her late 80s. Now you believe it, okay, I didn't need it, leave me alone. Let me go back to work. And she continued to do experiments. Until about a week before her death in the 90s. It turned she wasn't that lonely. People didn't think she was that crazy. And apparently her papers were ignored because she couldn't write, and her papers were incomprehensible. Nonetheless, she discovered all of this on her own. Staked her career on this. Most people thought she was crazy. Eventually vindicated. Really inspirational person. So, she discovers transposable genetic elements. She picked the right species to study, in the right organisms, which were plants. Think about it. You're an animal. And when things get tough, you can get up and run away, crawl away, or fly away. If you're a plant, you're stuck there. If you want to survive, you need to have something, some defenses. When a challenge, pathogen, climate change, etc. They have realms of their DNA, they move genes around, onto something novel and useful to get them out of the mess. Plants have inducible events of genes moving transposable genetic elements. Tend to induce them when the plant is at risk. A cellular stress response. They activate an enzyme called transposase. Enzymes tend to have ase at the end of the word, lactase, sucrase, transposase. You've got a defense on the part of the plant. Juggle some of its DNA. Make a copy of the gene and put it down somewhere else. And see if you stumbled into something useful. Later. They look at the same issue in animals, vertebrate, and mammals. We've got transposable genetic elements. E.g., 1. You're a scientist. You invented a pathogen or toxin. You've synthesized it and injected in a bunch of people. They get sick. You come back 2 months later. And their immune system will have made antibodies against that thing. So, how does the immune system come up with this variability for dealing with novel pathogens? Making antibodies that will recognize them. People discovered that splicing genes are relevant to making antibodies. Inducible transposable events. Making new types of antibodies. In a filtering process that goes on in the immune system, spotting them and fighting against them. That's a transposable event in the vertebrate immune system in response to novel pathogens. Like trypanosomiasis. Inflammatory disease. You get this parasite. Your body does this inducible trick. But the trypanosomes. A couple of weeks into it. They take away the surface proteins on their surface. And they juggle some of the relevant DNA. And come up with a novel version of it. So, as soon as you've got the antibodies online, they can't recognize the thing with those antibodies. So, you've got to start the process all over again. Coevolutionary races. It occurs outside of just the immune system and under some circumstances. E.g., 2. Transposable element. Predominant in primates. A certain cell type, when it's most mobile. Which is, the cells in your brain that are going to be making you new neurons. Neural progenitors cells. At the time they start proliferating and making new neurons. You're making new neurons, in a controlled realm of your DNA. You decided to change it, because you want to get interesting novel things that neurons can do. It means the cells in your body that have the greatest thing to do with making you who you are the least constrained by genetic determinism. Because when these cells/neurons are first being generated, they're changing more genetics than any other cell type in the body. That takes away the power of genes a lot in the nervous system. These transposable events make a whole lot of variability. Are these disastrous? In most of the cases, yes. By moving parts of DNA around. Making a copy of a stretch and moving it. It plunks down randomly somewhere else in the genome. So, you can have big macro consequences. E.g., 1. A world of promoters, of the actual gene. You've got an if/then clause. You're dehydrated. So, your kidneys work in a way that increases water retention. This is ridiculous. There is no if you're getting dehydrated promoter. There is no gene that's equivalent. There are networks, ways in which your kidney monitors other outputs in your body. So, we have a rough if/then clause. Now, you have some transposable events. The promoter world picks up and moves. And if you were dehydrated, part of the if/then clause floats around. It gets plunked down upstream from the genes that say go and ovulate. So, you've got an if/then clause. A promoter of that response to dehydration. And it turns on genes related to ovulation. This happens in seasonal mating. You're a species where 6 months of the year it's dry and 6 wets. Exactly the time you want to be having a baby. And you've got a 6-month gestational period. So, you want your body to know when it's dry season, that's the signal to make. Because you want to give birth during the rainy season. On the other hand, a species where you're pregnant for 2 weeks. And there you want to give as a rule, if it's the dry season, don't ovulate. Because I'm going to give birth to kids that are going to starve. Let's wait until I get a signal, that I'm totally wet and hydrated, then ovulate. You introduce novel if/then clauses. To how you would do season mating. Give birth given your gestation length, when your offspring is more likely to survive. And thus, passed on copies of your genes. E.g., 2. If/then element. A promoter. That can tell if an individual near me smells like me. Which shuts down transcription of things related to fertility. You don't mate with relatives, with individuals smelling like you. And now you've had a transposable genetic event. You plunk down the if it smells like me into the upstream of the gene that says cooperate. This is the start of kin selection. And if you have more promoters, If they smell a lot like me equals so cooperate a lot. If they smell only somewhat like me equals only somewhat cooperate. Sensory information about degree of relatedness. Possibilities of transposing genetic elements. Raises the possibility of moving parts of genes. Not just parts of regulatory elements. How you move a part of a gene. Exons, that's that modular construction of genes. Where if you get a transposase that comes and does its thing. Making a copy of a part, and moves the stretch around. You're moving copies of parts of genes, to create new genes. E.g. Basic mechanism of action of steroid hormones. Hormones like estrogen, progesterone, testosterone, glucocorticoids. They work this way. Steroid hormones can enter a target cell. And they bind to their receptor. They're not made of amino acids, have a different structure. Nonetheless, each has a distinctive shape and each type of receptor for a type of steroid hormone. Driven by its amino acid sequence. So, you've got a steroid hormone. Fitting into a receptor. As a result, is it activating this receptor complex? And on the other side, is a conformation which recognizes a promoter down on the DNA. A responsive promoter. You've got events going on in the outside world. Like reading a novel. And suddenly you're secreting certain hormones. And you're changing genomic effects. The environment is regulating genes like crazy. Requiring one part of the receptor, recognizing the hormone. And one part of the receptor, recognizing its specific appropriate promoter. Now, a transposable event happens. Steroid hormones. The hormone binding domain. And DNA binding domain. In steroid receptor genes, are in different exons. You clip the hormone binding domain. And stick it into a different hormone binding domain. So, you've made a different if/then clause. E.g. Glucocorticoids. Stress hormones. They suppress the immune system. These are steroidal anti-inflammatory, when you're taking non-steroidal. You're taking things that work like glucocorticoids on the immune system, don't have side effects. Glucocorticoids come in and bind to the hormone binding domain of the glucocorticoid receptor. And this translocate to a glucocorticoid responsive promoter. Now, a transposable event. What you've done, instead, is plunked down the hormone binding domain from the progesterone receptor. You've got an if/then clause novel. Instead of glucocorticoids suppress immunity, it's progesterone suppress immunity. Progesterone, progestational. So, you suppress your immune system during pregnancy. So your body won't eat your baby. Don't have an immune reaction against it. They decide this thing belong here. Instead of this thing have invaded my placenta. Related to the imprinted genes. Male-derived imprinted genes making for a more potent invasion into the placenta. As a consequence, you're immune suppressed because you're pregnant. When give birth and stop being immune suppressed, the progesterone disappears. Your immune system comes back to normal. But your immune system might be so wiped out of control. What if recovers from pregnancy immune suppression? And instead of coming back to normal, it shoots into being more active than it should be. You can get autoimmune disease in the onset of postparturition period of women, like lupus. So, by doing transposable event, moving one exon around and sticking it to another new exon, you've made up a novel if/then clause. We are away from gradualist world of micromutational of one protein working differently. You have the capacity to invent novel proteins. Through splicing enzyme changes. Through transposable events. A novel network. With mutations and promoters. Mutations and transcription factors. Is going to be major changes. Sometimes, a gene will be duplicated. And create an extra copy of the gene. Gene duplication. Even a larger expansion of the number of copies of a gene. Or duplication of a whole stretch of genes. Copy number variant. From one extra copy of one gene to massive duplications of stretches of chromosomes. Like schizophrenia. Involves mutations in copy number variants. Not a mutation on one base pair, or in one transcription. This is extra copies of genes sitting there. In some cases, the second gene can function as a backup. Something goes wrong in the first. The second do his job. That occurs in some subsets of Alzheimer's. Or, number of copies of the gene you make. Has something to do with how much protein you make. E.g. compare Japanese populations with Western European. On average, Japanese have multiple copies of a gene. That makes an enzyme related to carbohydrate digestion. This isn't a populational difference in a DNA sequence. It's the number of copies of the gene. Also, with the duplication, you can experiment with one of the copies because the other will take care You get faster evolution with genes that you have duplicated, where one is more free to do more dramatic movement. And you can a great use without sacrificing the initial use in the process. Joe Thornton. Evolution of genes for steroid receptors from ancestral genes. Two different genes for two different types of steroids receptors were once duplicates of the same gene. One could float, and stumbled into something useful, while the other one held into place. This helps explain to people who attack the notion of evolution. Irreducible complexity. That evolution can't exist. Because what good is half of an eye? You've got to have intermediate forms. Evolution couldn't have produced an eye in one mutation, one generation, and thus, would have to be in a series of steps. Evolution can't exist. In this case, you get a demonstration. By having extra copies of genes, one free to be evolving. You don't have to have a rapid transition from one to the other. One moving, one doing the job. Until it comes up with the shape of a receptor that can bind a hormone that stumbled its own way into existence 10,000 generations earlier. Which because it was duplicated. Where one copy was now of a form where there was no receptor for it. Until it happened to stumble into that. Duplicated genes describe intermediate states where don't have half an eye. But instead, you have the pieces ready on place for when one thing pops up which completes the picture. Russ Fernald. How eyes evolved from a single layer of cells on the surface. You can have half an eye intermediate form. We've got all the pieces in place. Changes in splicing enzymes, promoters, transcription factors, transportable elements, number of copies of genes, number of stretches of genes. What wind up being the consequences of this? Mess with a single base pair, one single gene. And get terrible problems. PKU. Now, with these macroevolutionary changes, change one single base pair. You change more than one different gene. You have one single base pair change. And if it's an exon used in a lot of combinations, you've now produced mutated versions of a bunch of proteins. One change in the transcription factor. And you've invented an entirely new network of expression. But, it's really unlikely to get positive results out of it. This is a stabilizing mechanism for equilibrium. Most of these macro-mutational changes are going to be bad news. In other words, there is stabilizing selection against macro-mutational changes. We get an evolutionary bottleneck. Circumstances of severe selection of a tiny subset of traits. If you have that trait, it doesn't matter how much of a network you've changed. You're going to be one of the ones who comes through. And everybody else does not. Throughout the history, there are records of selective bottlenecks, where 1% of the population comes out because of some rare trait, which carries all the macro consequences. E.g., bottleneck of selection of cheetahs. About a couple thousand years ago, they almost disappear because they're so genetically similar to each other that you can transplant tissue onto another one, and there won't be rejection. All cheetahs are closely related descendants. A tiny remnant population, highly inbred. E.g. to hominid evolution. Almost disappear because of glaciers or comets. Only a small subset of individuals with some traits that have been up until then neutral or not really useful, because of big macro consequences. Only the ones who had it, came out the other end. Here we have circumstances of rapid change. Punctuated radical change. To make conceivable punctuated equilibrium. Intrinsic in it, are two critical things. 1. The majority of time. Make a change which changes the network. It's going to be disaster. The majority of time stasis. 2. The ability to have massive macro changes. Where in periods of selective bottlenecks, you'll have rapid changes. Long periods of stasis. Very rapid change. Under some circumstances. Ways that it could be done. 1. Micro-mutational changes, changes in the function of pre-existing proteins. Macro-mutational changes, invention of new proteins, networks, if/then clauses, serving different functions. Differences in the genome between humans and chimps. When you look at the micro vs macro differences. Immune system. Genetically have to do with micro differences. Humans are more resistant to tuberculosis. Chimps are more resistant to malaria. It's the basic nuts and bolts of the immune system. This is within a realm of gradualist change. Development. Have to do with macro. One little difference. You get an organism that looks so different from a chimp. What else in terms of resolution? Fossil histories support punctuated model. Nonetheless, some follow gradualist models. But mostly punctuated evidence. It's impossible to see gradualism going on. Because it's gradual, it's hard to spot whether are incremental or rapid. E.g., people in Chicago Have all sorts of carcasses around from rats killed, in the 1880s. Researchers looked at the genomes. Compared to rats taken these days off Chicago. A lot of the genome has evolved. Or finches, in Galapagos. Gene distributions are changing. Gene distributions for traits. Like the size of the beak, and thus what kind of food you could break into. In response to environmental shifting. Evolution of resistance to diabetes. Comes in two forms. 1. Juvenile onset diabetes, where you need insulin. 2. Adult onset diabetes, a lot of calories, obesity. People studying people that have had rapid shifts from traditional diets to westernized diets. Native Americans in Arizona. Pima Indians. Half live in USA, rapid changes in diet. The other half in Mexico, less changes. People in Arizona. 90% diabetes rates by the time you're 30. So, you've got people who are most vulnerable to diabetes. When giving them a westernized diet, they're leaving fewer copies of their genes. But in the last decade, the diabetes rates are beginning to go down. There has been the selection that the folks who have the most efficient metabolisms leave more copies of their genes. And this is within the course of a century. Soviet Union. Siberian silver foxes. It's highly valued to have clothing made out of their coat. Someone started breeding them for tameness. They captive baby foxes, most of them feral and crazed. A subset of 5 to 10% of them. To get closer to the calmer foxes in the human's proximity. Only let those ones breed. Now take their offspring. Only take the new 10% calmer and so on. Took about 35 generations. To get foxes as tame as dogs are. So, he's breeding them for only one trait, a behavioral trait. And when you spend 35 generations, the ones who are most able to function in the role of domesticated animals. It's not just the behavior that changes. Also, their appearance, variety in their color, adrenaline and melanin hormones are chemically connected, so they change together, neurotransmitters dopamine and noradrenaline which control behaviors. Ears became floppy. Started to bark. Playful. And no longer afraid of people. Will wag its tail at you. Lick your face. We got dogs the way they are. Because people just selected them by tameness, and evolution did the rest of the job. Basically, you're breeding a wild animal to be tameable. Breeding them for infantile traits. Ironically, the code changed. Piebald coloration pattern. You can't get the domestication without that happening. Ironic ending. Nobody wants to wear clothes that look like that. Also, consequential. Shortened life expectancies. Antibiotic resistance in various bacteria. Evolution occurring rapidly. And at some cases, it's going to leave a different sort of skeleton. Changing how good your kidneys and pancreases are dealing with westernized food. It's stuff that no punctuated equilibrium paleontology would ever be able to pick up. At the end, all have means of having different types of evolution happening. Conclusion. Microevolutionary changes, the immune system in chimps vs humans. Macroevolutionary changed, development. They could be going on at the same time. In a resolving way. You've got evidence for punctuated change of some trait. And single traits don't evolve at a time. They come in packages. So, there's some other punctuated thing going on. Meanwhile, you've got other trait moving, evolving in a punctuated manner. And you put enough of punctuated events happening. And on a whole population level. You've just invented gradualism. Next lecture. Trying to make sense of the genetics of behavior. Stories built around individual selection, kin selection, group selection. And come up with the best story around to show me something more predictive, and you win. Next lecture. Showing actual genetic changes over the course of time. Patterns of evolutionary change. Will those code for what consequences? For understanding the evolution of behavior, competition, reproductive strategies. Then, switching to behavior genetics. What's going on when somebody gets adopted? They share a trait more with their adoptive parents? Or their biological parents? Identical twins vs non-identical. Identical twins who were adopted into different families at birth, and put them back together.